Bismillahirrahmanirrahim. Students, there are different products of the sustained control release in which there is a mechanism-based classification of the systems. Number one is known as dissolution control system, in which we have controlled the dissolution rate of the water-soluble drugs. Second one is a diffusion control system, in which we have controlled the diffusion parameters of a lipophilic drug, or we have controlled the diffusion parameters of a hydrophilic drug when it is coated with a polymeric membrane which is lipophilic in nature. Third one is combined system known as diffusion and dissolution controlled drug delivery system. It have the both properties of dissolution and also diffusion. So first it will start diffusion and then it will start dissolving. There is a next mechanism which is known as chemically controlled system, in which we have prepared a bond between the drug and a polymeric material. Until unless a bond is broken by some enzymes or some biological conditions, this delivery system cannot deliver a drug. So any bond which is dependent on a chemistry of the formulation, it will be known as chemically controlled system. There will be multiple bonds which are formed between drug and polymer. Each molecule of the drug and polymer will be bonded by these bonds. So these bonds, as these will start breaking, it will release the drug, it will start releasing the drug. Ion exchange resins have different ions in a resinous material, in which it can demonstrate ions depending on the charge of the external environment. So resin will allow the ions to move from the resin into the charged environment to make them neutral, and will adjust its surface by taking other ions. So the drug can be delivered by replacing these ions. In a hydrogel system, it is also a combined diffusion and dissolution control system, which is known as swelling control system also. So here the polymer release the drug when it is swollen, or it has taken an area or volume which is greater than the administered volume. There is a system which depends on the osmotic pressure which is generated within the system. So higher the osmotic pressure, higher the drug delivery. Lower the osmotic pressure and lower the drug delivery. So there will be an osmotic agent that will control the release depending on the osmotic pressure. There are also magnetically controlled system, in which we generate a magnetic field. In that magnetic field, the drug also synchronized with the cellular environment, which have a magnetic environment. So when a synchronized magnetical environment is developed, it can deliver a drug, or it can release the drug from the system. So for the first system which we discussed lastly was the dissolution controlled system. It has dissolution control system, as by name we will make it by decreasing the rate of dissolution. So there are three approaches to develop such kind of a drug release. One is a chemical approach. Chemical approach means you have formed a bond that will decrease the rate of the drug release. For example, you have chemically developed a bond between a salt forming agent and the drug. So that salt is of lipophilic property, means it is a sulfate, it is a ammonium salt, or it is a further category of acetones or ketone salts. If these are such kind of a chemical association, then its drug release can be decreased. Second approach could be a coating of the drug with slow dissolving polymeric material. Coating could be of two types, in which we can coat a drug with alternating layers of the polymer, or we can coat the drug with thickness of the polymeric material. So alternating layers of the drug at a control rate coat can be demonstrated, or a varying thickness coats can be incorporated to form a slow dissolving drug product as a dissolution control system. Third one is a matrix that have a polymeric material that allow the drug to be eroded slowly. or to be removed slowly. The matrix is actually a homogeneous blend of the drug and the polymeric material. So rate of penetration of the dissolution fluid in the matrix will determine what kind of the dissolution and release will be from the matrix system. If we can solve the kinetics of the dissolution control system, it depends on the Noyes-Whitney equation. Noyes-Whitney equation is directly proportional to the area of the formulation which is exposed and the concentration gradient maintained. If the concentration gradient exists and area is available for the drug release, there is a continuous change in the concentration with the relevance to time. So dissolution rate can be changed with area and concentration gradient. With Kd as a diffusion rate, dissolution rate constant, Kd is actually equal to d over h. D is the diffusional coefficient and h is the thickness of the membrane. If we can make the system which is constant at surface area level, it has a constant diffusion coefficient. There is no wear and tear of the polymer. Our diffusion coefficient remains constant. Diffusion layer thickness remains constant. It doesn't change, and concentration difference exists for the whole time of the drug release. Then there is a possibility that this drug release can be independent of the concentration, and it can be of controlled release. But here, dissolution, if we say this is a dissolution control system, so it will always depend on the area which is present to be dissolved. So whenever one area will be dissolved, the next area will be different from the previous one. So area will change, so the concentration will change. As the area changes, concentration changes, so it will depend on the first order kinetics, means concentration system. So it can be better demonstrated by replacing the concentration with the weight basis, weight of the particles. So as the weight can be determined easily, and particles can be on the weight basis can be ensured that they are in a sync condition. So we can rearrange that equation into of the Noyes-Whitney equation into cube root law, Hixon Crowell Cube root law. W naught, that is initial weight, and W, which is the weight remaining at time t. Cube root of the W naught minus cube root of the W weight remaining at time t is equal to Kd, which is a diffusional coefficient or diffusion constant, and time t. If the weight change, which is cube root weight, it remains constant, then it will be dependent on the time. If there is a slight change in the weight change during the kinetics, it will not depend on the time, or it will be varying on the basis of weights. So weights are actually concentration, so it will become first order kinetics. So normally, if there is a linear relationship between change in weight to time, then it will be controlled release, otherwise it will be sustained release. A second system, which is known as a diffusional system. Diffusional systems are the systems in which drug release is dependent on the diffusion of the polymeric material. So this can be achieved if we develop a formulation by incorporating it in a polymeric material which is insoluble, or which is a inert membrane polymer, and it is not hydrolyzed, or it is not being metabolized by our biological system. So such kind of the membrane barrier will serve as a whole, as a constant surface area. So that constant surface area that can be put in the Noyes-Whitney equation will result in the constant drug release, which is controlled release. So if we are preparing a system which is only dependent on the diffusion of the drug from the system, so we will require a polymer which will be lipophilic in nature or insoluble in the biological condition. So that polymer will ensure that its surface area doesn't change, or thickness of the membrane from which the drug is releasing doesn't change. If it is doesn't changes, H doesn't changes, diffusion coefficient doesn't changes, area exposed doesn't changes, then the concentration gradient is maintained always, then the drug release will be controlled release. There are two types of approaches of the diffusional system. One is a reservoir system and other is a matrix system. Reservoir system is the same in which we have discussed in the dissolution system, that reservoir system will be a coating system. There we have coated two types of the coatings. One is a different thickness of the coat and one was the alternating layers coat. Here reservoir system, we can coat a polymer on the drug molecule that will remain intact throughout the delivery system, through the time of the drug delivery. So if it will remain intact throughout the drug delivery time, it will serve as a reservoir. So reservoir system are considered coated system. Matrix system are the second example. Matrix are homogeneous blend of the insoluble polymer with the drug material. So that matrix will have a drug which is dissolving but polymer which is undissolving. So drug will penetrate through this matrix to the dissolution medium. So rate of penetration of the dissolution medium will draw its kinetics towards sustained release or a controlled release. Starting with the reservoir system in which there is a central core material which is active ingredient, and there is a polymeric membrane around the core material. If we can draw a sketch of cross-sectional sketch, we will find that there is a internal area which have a concentration of the drug as C0. There is a membrane area which have a concentration which is Cm, membrane concentration, and there will be outside the membrane which is biological fluid. Here the concentration will be Ct, a time concentration. So if we can calculate its kinetics, here the concentration which will be from the core material dissolved will penetrate into the membrane, and the concentration at this junction will be Cm naught, zero concentration, Cm0. It will have to travel the distance of this membrane. So the concentration which will be attained after traveling this distance, if this distance is a d, it will be Cmd, concentration of the membrane after traveling distance d. Here the concentration of drug in the membrane at zero distance and it is at d distance, and the drug will be released from this membrane surface into the dissolution medium, and the dissolution medium will have a concentration which is concentration after passing this distance d. For Cd, it can be denoted as Ct also. If we can calculate diffusion parameters, so diffusion parameters can be calculated from the Fick's law of diffusion. Fick's law of diffusion is the flux of the drug which is directly proportional to the change in concentration per unit distance traveled. So it can be equated by using a diffusion coefficient d. Diffusion coefficient d actually represent area per unit time. So diffusion coefficient is d, which is area per unit time. dc over dx will represent the change in concentration with relevance to distance x. C change with difference to relative x. So here there is a the flux will depend on the diffusional coefficient. That means that the area which is exposed at a specific time period. If the area remains constant, then this flux will remain constant. But as area changes, normally concentration based areas are changing, so d always decreasing. Diffusional coefficient is actually decreasing. So to represent that decreasing, we are actually having a sign which is minus sign. So flux is always towards the downward side. So J flux is equal to minus d dc over dx. Here we can add those kinetics which we have solved for the membrane. If the drug was in the core material, it will be known as C0. If it is in the membrane at the first junction layer which is attached to the core material, it will be having a concentration Cm0, and when it will travel the distance d, it will have a concentration Cmd. and the drug will be released out of the system. So K can be calculated from this C naught, which is inside the core, inside the polymeric material, core material, to the concentration which is outside the core material. So rate constant will be dependent on the Cm and Cd concentration. The concentration which was available at the initial membrane concentration, which is actually C naught, behind our C naught. Cm naught divided by Cd at x is equal to 0. And also K depend on the concentration which is available, means concentration gradient. It will be depend on the concentration gradient, which is Cmd, the concentration of the drug which has traveled the distance, d divided by Cd. It will be at x is equal to d. So K can be replaced with for the diffusional coefficient in the flux. Diffusional coefficient which was actually changing in the negative direction always. So we can replace it with this K, which will be actually diffusional coefficient and will be a ratio of the drug concentration in the membrane to the bathing solution, or the ratio of the drug in the core to the bathing solution. So alternatively, because there is a membrane existing, so always K will be a diffusional coefficient and it will be the ratio of the drug concentration in the membrane, in the bathing solution, from the core membrane to the bathing solution. So we can replace this, we can add this K into the J flux equation. So it will become minus dk dc over d. That is the equation of the actually diffusion. So if we can add a equation which is a diffusion equation, or equate it into a diffusion equation, so the flux will be converted to a diffusion equation. As a diffusion equation also have dk dc over d. If we add area in that, so area in there added can be converted, flux can be denoted as diffusion equation. So diffusion equation is dmt over dt is equal to, for equating minus sign, we have added area. If the area will remain constant, there will be no negative sign. So if area is added here, so it will become the amount of change of the drug per unit time as a constant rate. So it will be dependent on A, K, D, dc over d. The equation can be actually demonstrated for all kind of the systems which are formulated, all kind of the geometries which are formulated. For example, we can formulate a round delivery system which is known as spherical delivery system. So for spheres, it can be written for spheres. We can formulate it into a tablet-like or capsule-like shape or pallet-like shape. So these shapes are cylinders. So cylindrical equation can be written from this equation. And we can formulate into membranes, into slabs, into longitudinal or flat surfaces. These flat surfaces are known as slabs or patches, जिसको हम दूसरी लैंग्वेज में कहें transdermal drug system. So transdermal drug system can be developed as slabs. So the equation can be written for all these three geometries: spheres, cylinders, and slabs. So to achieve a zero order drug release, if area of the drug always remains constant, if it will be a non-dissolving polymer, the area of the drug from which the drug is releasing, area of the system from which drug is releasing will remain constant. So A is constant. If A is constant, there is no change in the polymeric geometry. So D will remain constant. Diffusional coefficient will remain constant. And the concentration, if the concentration gradient is available always, there will be change in concentration always existing, and K will remain constant. And D distance, if the distance remains constant, it will be a zero order release. There is a problem with the distance. As it will be a reservoir system, there will be no change in the distance, as it have a single membrane around the reservoir or around the core material. So the single layer will serve as a distance d. But in the matrix system, it will be changing, as matrix have the uniform blend, homogeneous blend of the drug and the polymer. So drug that is in the extremities will have a lesser distance to travel to the bathing solution than the drug which is present in the center of the matrix. So there will be distance, there will be a variable existing known as D. So depending upon D, the drug concentration will not be constant. So zero order is possible for reservoir system, zero order is not possible for the matrix system. So it can be as a summary, a reservoir diffusional system could be drug is surrounded by a polymeric membrane. Number one, zero order release is possible as area remains constant, diffusional coefficient remains constant, distance remains constant, and the drug concentration remains existing always. So zero order release is possible. So that can be actually release rate can be varied depending on the polymer. If the porosity is controlled, tortuosity is controlled, we can extend the drug release from a specified time to a extended time period. Disadvantage of the system is that system, if the polymer is not dissolving, so system will remain at its site even the drug is exhausted from the system. So the system could be physically removed from the site where we have administered it. So this is the disadvantage that we have actually placed a system. It is exhausted by releasing the drug, but still a ghost polymer is present over there. So to remove the ghost, it is a system which is having a problem that a polymer will exist at its place. And the polymer can be toxic. So polymer should be degrading. First disadvantage could be covered if we use a degrading polymer. And if we're using degrading polymers, its area will not remain constant. So zero order release is not possible. Secondly, as the drug release is dependent only on the porosity of the polymeric material, so the porosity will not allow the high molecular weight compounds to be released from the polymeric material. So it will be difficult to release high molecular weight compounds. Thirdly, if we are using such kind of a polymer which is not degrading biologically, so it will be having a higher cost. We will have to develop a sophisticated coating system. We will have to adjust its pores very specifically. We have to develop a polymer very specifically. So its cost will be increasing. So as the TDDS system have a higher cost, there could be a potential toxicity of the system as we have added multiple doses in a single system. So potential toxicity will be if the system is failed or it can be administered by breaking the tablet. The system can release high dose or a toxic dose that can cause potential toxicity of the system. Okay, now come to the second system, which is known as matrix system. Matrix, as we know, is a homogeneous or uniformly blended drug with a polymeric material. So an inert solid vehicle in which drug is uniformly suspended will be known as matrix system. As we have stated that diffusional system are having a polymeric material, a solid polymeric material, which is insoluble. So if it is insoluble, it can only release the drug depending upon its pore size. And if it is releasing depending on its pore size, it will be controlled by diffusion process. And if the polymer changing its porosity, not changing its surface area, but porosity is changing or adjusting its surface area to a surface area from which it can release, drug will be releasing from that swollen area that will remain constant, or drug will actually start, or the polymer will actually start eroding. So the surface area can be converted into erosion surface area, eroding surface area. So matrix have a diffusion process alone, it will be control release. Have a concomitant swelling system in which a drug is releasing from a swollen area and the area doesn't changes, it will be control system. Or matrix could be a erosion process. If it is eroding, every time surface area will be decreasing. So a constant drug release is not possible from erosion process. So if we are using an erodible polymer or a swellable polymer which erodes after swelling, so the release kinetics will be complicated. It will not be depending upon that. Similar diffusion equation, a new equation could have to be developed for erodible, swellable polymeric matrix, not for the only diffusible polymeric matrix. For only diffusible polymeric matrix, we can adjust it into the same diffusion equation. But if it is changing, there will be a changing boundary, there will be changing area, and that front which is eroding or which is swelling, multiple fronts will exist, a swelling front and eroding front. That's fronts which are eroding, which move opposite, in the opposite direction or in the same direction. In the both cases, it will result in the different rate of diffusion. So drug will depend on the swollen front, eroding front, or the diffusion front that have developed a diffusion front. So these fronts will demonstrate how the drug will remove from the matrix system. If the drug is present in the small percentages, so matrix can protect the drug from rapid dissolution, as the drug will be held in a polymeric material. So polymeric material will swell and then will allow only drug which can be diffused from the pore size. It will control the side effect of the normally our reservoir system, in which we said that if the reservoir system is broken, its membrane is broken. A broken membrane will result whole the drug to be released at the same time. But if a matrix is broken, it will have the same control with the half piece of the matrix, or it will have a same control with the fourth piece of the matrix. But as we will decrease the size dependent diffusion parameters are also different. So depending upon the size, volume will increase. So as the volume of the diffusion increases, it will have a more penetration power to the small sized matrix system. So drug release will be increased, but instead it will be released immediately, as in the case of the reservoir matrix have a superiority than the same type of reservoir system. So in the matrix, there is a drug dispersed in a polymeric material. When the solvent will penetrate, for example, here the drug is in the darker orange circles and light orange color is with the polymeric material. When the solvent is penetrating at time 0, it's look like this type of a matrix system. When solvent is penetrating in the matrix, it will dissolve this drug molecule, and drug molecule will be diffused from this polymeric material. It will come out. For example, at time t, a surface bound drug or entrapped drug is actually released from the polymeric matrix, and then next drug molecule will be dissolving, and that will be diffusing out of the polymeric matrix. This process will continue until a whole drug will be evacuated from the matrix, and this polymeric ghost will remain intact. So it has again the same problem which was with the reservoir system, that polymeric ghost remains within the system. So matrix for a drug release which is dependent on the matrix can be controlled by having some assumptions. For example, there are different scientists which have assumed that if such kind of condition exists, matrix will be a zero order release. But if such kind of conditions doesn't meet, then it can depend on the equation which is a concentration independent but not a zero order. So there is one scientist known as Takiro Higuchi, which has mathematically developed an equation for the matrix system, or derived an equation for the matrix system, and based on the four assumptions, the following four assumptions, which are: number one, if a polymeric matrix can demonstrate a pseudo steady state known as a constant drug release from the system equal to the amount of the drug which is evacuated from the body. it will be known as steady state. A steady state concentration could be maintained. If the area is changing, it will be pseudo steady state level. So at least for our matrix, if it is releasing at a pseudo steady state, a pseudo steady state is maintained during drug release, then this equation can be derived. If that pseudo steady state is not maintained or steady state cannot be generated, then this equation cannot be derived. Secondly, the diameter of drug, which is a drug particle which have to diffuse out of the polymeric matrix, should be less than the average distance, which is average distance of the polymeric matrix in which it have to diffuse out. So diameter of the drug particle should always be less than the average distance of diffusion through the matrix. Secondly, this is the second parameter. So high molecular weight drugs which have the diameter of the drug particle more than the average distance will be restricted within the matrix system until unless it is eroding. If it will be eroded, then that drug can be evacuated from the system. Otherwise, that drug cannot be evacuated from a system. Number 3, a bathing solution or sink condition should always be established. Sink condition means the solvent which is to be penetrating into the matrix should remain always intact, or the delivery system should be dipped within the solvent in which it is eroding. If that sink condition is not maintained, it cannot release the drug, and Takiro-Higuchi equation cannot be assumed for that drug release. And fourthly, diffusional coefficient of the drug should remain constant. Means there will be no change in the characteristics of the matrix. For example, matrix is not eroding, matrix is not swelling. If it is not eroding, it is not swelling, then diffusional coefficient of drug will remain constant. Then this equation can be derived. So there are 4 assumptions on the basis of which Takiro-Higuchi derived an equation for the matrix system which is independent of the concentration, but not on the concentration, so it is not zero order but near zero order as it is independent of the concentration. So there are 4. A pseudo steady state level should be maintained during drug release. Diameter of the drug should be less than the average distance of the diffusion through the matrix. Bathing solution should provide sink condition all the time. Diffusion coefficient of the drug should remain constant. For the kinetics of the Higuchi, if there is a polymeric matrix in which drug and polymer is homogeneously blended, so there will be drug with a polymeric ghost. Polymeric ghost is that polymeric area which have exhausted from the drug contents. For the first time, it will not having any polymeric ghost. It will be a matrix. But after the first release, it will be having a polymeric ghost around the drug particle. So drug have to travel this distance. After the first release, drug have to travel always this distance or greater than this distance to be evacuated from the system into the dissolution medium. So here, if the first line is this one where x to travel by the drug is 0, and then the last membrane is with the distance dh or with the distance h, if x is h. So the concentration can be written as, here the concentration which is CS, saturated concentration, which is saturation of this polymeric matrix, and here will be concentration which is CT or concentration C, which will be out of the membrane side or into the dissolution medium. So it can be written as change in the concentration, change in amount of the drug per unit area, dm over dh. This will be equal to the concentration which is initially available in the system multiplied by the average distance it have to travel minus half of the saturated concentration. Because every time when there will be a distance to be traveled by the drug, it will be converted double. Distance will be becoming double than the actual distance, as from the matrix it will be going, drug will be going centerwise, and the polymeric ghost will remain constant. So it will having a distance which is always double than the first one. So this can be written as half of the saturated concentration, mean half of the saturated concentration will be remaining always. So C naught minus Cs over 2 into dH. This can be written as Higuchi equation. From here we will start in the next lecture. So until now, there is end of the first lecture.